Usually,
So that would be too much.
I mean, there's balance. If you are putting more than one paragraph, that I think it's more. But like, I added one paragraph of like more background information. She said, "Oh, it's really better than last time." So probably one paragraph less than 1 page should be sufficient.
Okay.
It depends on like how like you go. Like if you go too tangential, it's not.
Yeah. It's just like a.
Yeah, like. Or like you can even mimic the introduction of the paper you're actually looking at. Like this is thing we're looking at, and this is actually carbon issue. So that's why you try to see as well.
So why are you even interested in examining?
Or that bacteria is causing loose infections.
It's kind of multiple things.
Multiple things. Yeah, you can list that out too.
Yeah. Like it helps fight against like animal toxic papers in terms of that and bacterial vaginosis. So we can just
that's important
talk about that
in that term. So we can probably like say this is important because this is this. Like, you just abruptly introduce like all the activity this bacteria does is because of to fight the environment. Like yes, that is important, but what do you feel about?
Okay.
What I feel like I want to know.
Okay. And then I'll talk about it like specifically.
Yes. And that because all these activities like actually try to fight against these like external factors, which is something.
So should I put that before my before what I already have?
Or like you briefly explain it and then like put in the second paragraph. That's what I did. So like I'm doing this like a protein, right? Oxygen protein, carbohydrates, everything before that. And then I'm actually like looking into protein, other proteins that is helping that central protein. But like because this I'm interested in this other protein. It's important to talk about central protein because you cannot just talk about something. Yeah, this helps this one. Then what is this? Like what is it for? So I explain I'm interested in this other protein, but firstly I'm discussing central part so that you better understand what's happening.
Okay.
I'll eventually do another like draft session with stuff for resume because my paper's not done.
Yeah, I saw
it's
that.
It's not best draft. Yeah.
I was like, oh my god. I was like, I would not want to read your paper. I would not want to read your paper. I'm not gonna lie to you. And then I was like, how many pages is it? She was like, oh, it's 5. I was like, what? And then she says, but it says it's not done. I was like, oh, that makes sense. It's like, I can see you having like 8 pages. You're gonna have 8 pages. Hopefully you have more.
And at the same time, it's like too much yet. And obnoxious terminology as much as possible, but Dr. Reverend said, no bueno.
She probably doesn't want to read that.
No.
Yeah, then when I introduced some terminology, she didn't even know what, she literally said that. She was like, I don't know what this is.
Right.
So I explained that in the paper a little bit.
Or even what I did is eliminate. That's how my paper reduced from 8 pages to 7, 6, 5.
Yeah, I felt like me too, because I took a few topics out and she was kind of like, so Larry.
Right. Yeah. I mean, if you can give a rationale, like rationalization is good, but at the same time, yeah, probably you don't need it. It's just still junior paper, not the actual academic paper you probably something you don't probably submit on.
Yeah, that's why I don't really. But I'm like, I care. I don't really.
I wish I could can be on my resume.
Like publishing?
Yeah, not this, but if I, just more details on it.
Yeah.
But,
We'll make it through.
we'll make it through.
It's kind of nice. We're going to have the peer reviews done by next week.
Yes.
That's just too much.
I got through this success.
I was kind of confused. I was surprised, I guess, that she only wants a half a page review.
I mean, there's a point, like, you can pick people's, like, if there are too many, like, just can't do it once. And just, she's probably trying to keep it, or like, there are 2 reviewers.
Mm-hmm.
I think the point point is instead of like one people rip off the paper, like give people like have two different perspectives on life. Probably she's like experimenting with our
She's gonna be late.
way.
She's at the
Like,
Thank you.
did you see Dr. Salcido yesterday?
No, that
She
was
showed
the
up.
senior center.
Yeah, but like before that, I showed up to the room. It was like she would have oh, she's like experimenting with us. We're guinea pigs.
Yeah.
Which is understandable. Maybe sick, raining too much.
Wait, you said that about the attendance? I thought we were just gonna get extra credit.
Oh, no, I swear he said 50 points of your attendance grade. I swear he said
That's
that.
bad. That's a
Nobody
whole.
was there.
Do those without saying all that. So
I swear he said that.
No, because our attendance is I think he can, because there's only like 2 days you have to take attendance or something.
right, but
When he told us that our attendance is our curve.
Yeah. So like if you don't show up.
You don't show up, you don't show up.
yes, you don't show up, you don't show up.
Gosh.
Alrighty guys, we'll go ahead and get started. So today we're going to be talking about food microbiology. This is most likely going to be part 1 of the lecture, and we'll continue on next week on Monday. The chapter in the textbook is chapter 16. That would have all of the content. So the microbial ecology of food. So basically there are microbes that contribute to having good food, and they can also contribute to having, spoiled food as well. And they can also contribute to even uglier consequences like getting sick and, things like that. So we're going to definitely talk about all of these things. And so microbes are involved in producing food and preserving food, so we need microbes for those things. But then microbes can also cause the loss of the appeal of the food, the loss of the positive flavor, and having a smell. And then the ugly microbes also can cause food poisoning and illness and getting sick. So you've seen this before. One of the ways that microbes get their energy is through fermentation. And so some fermentation products are going to differ depending on the type of microbe. And so there's a lot of different organic compounds that can be fermented. And so we've talked about butyrate, acetate, propionate, lactate. So depending on the microbes, that's kind of what differentiates what's being produced. But microbes are important because they produce these acids. So we've talked about them in the context of gut health, and so today we'll talk about them in the context of food fermentation. So microbial fermentation can be used to make and preserve food. So this is obviously something that you guys are all very familiar with as college students. To make beer, we need microbes and their fermentation process. To make pizza and cheeses, breads, wine, ciders. So all of this really has to do with microbes. We've been doing this for hundreds, if not thousands of years, especially for beer. So fermentation of these microbes, that's a really important method of food preservation, so to actually preserve the microbes. And so the acid produced as the fermentation reactions proceed is what makes, us be able to keep the food preserved. And so, for instance, in yogurt, cheese, and pickled vegetables, it's that acid that's produced in the yogurt, cheese, in vegetables that inhibits the growth of organisms that can spoil the food and foodborne pathogens. So those fermentation products, those acids are really what are important in preservation. And then in addition to that, we know that microbes also produce their own antimicrobials. Those are called bacteriocins. And so they form those and can also lead to food preservation by inhibiting the growth of other microbes. And so this is, inherent to the bacteria. They produce these acids and bacteriocins, but there are also other outside factors of historically important preservation methods that we've utilized, and that includes salting, drying, adding various herbs and spices, and then canning, or it's called pickling as well in other contexts. Any questions so far? Yes.
Can you explain the bacterias?
Yeah. So microbes, they naturally will produce these antimicrobial peptides, and basically they are microbes, but they produce antimicrobial peptides that we call bacteriocins to inhibit the growth of other microbes that we don't want in that on the food. So lactic acid fermentation by lactic acid and acetic acid producing bacteria. These are really important for pickling and making cheeses. So the taste of yogurt, pickles, and sharp cheeses, and even some sausages are due in part to production of lactic acid and or acetic acid by these bacteria. So they also produce other molecules and things like that, but it's this lactic acid and or acetic acid that's produced, and it's produced on the actual vegetables, or like on the pickles here and on the cheeses, and it's produced by microbes that are inherently on them. And so we've heard this before as lactate or acetate, so now we're talking about it in the acid form. So cheese, yogurt, kefir, and other fermented milk products. So a number of products are made from milk, as we all know. And so milk, in theory, it's actually sterile in the cow's udder. There's absolutely no microbes at all, but it can rapidly become contaminated during milking and handling. And so there are lactic acid bacteria. So lactic acid bacteria are usually commensals. They're good for you, and they help to keep things, in check, and they are producing lactic acid, for instance, but they generally reside on the cow's udder, and so when they're milking the cow, that will cause contamination to that sterile milk. And so the aesthetic features of milk change due to the production of acid and proteases and other things and flavor compounds. And so it's the fermentation processes of the microbes that are in the milk that then can change the characteristics of the milk. And so this then can cause the milk to coagulate or curdle. That just means it becomes chunkier, and then it can sour the flavor, it can thicken the liquid. And so kefir is one of those fermented milk types of drinks. That's just an example of it. That's similar to thin yogurt. It contains live microbes, so it contains live probiotics, and it makes a great smoothie mix. But this is one of the examples of, just using milk and then just making it a little bit thicker, but not too thick, to kind of make it more of like a thin yogurt. But the point here is that although milk from the udder is sterile, you don't drink raw milk, because it can more than likely will get contaminated in these ways. So there's germs that can come into the milk from the animal's poop. The skin, like we said, the udder can have these lactic acid bacteria on them, or they will have these lactic acid bacteria on them that will inherently get into the milk and contaminate it. Germs in the environment can get in the milk. The udders can be infected, so mastitis is this term that describes an inflammation and infection in the mammary glands of the udder, and so that can definitely, cause infection. The animal can have a disease that's obviously not readily observed by the human eye, and then there are insects, rodents, and other small animals that can get into the milk. And so an actual milk processing plant, that can be unsanitary, at the entire place. And then on top of that, cross-contamination from the dairy workers on their own hands and their boots and things, they can, come in contact with, that will come in contact with the milk and contaminate it as well. So, there's a big push, obviously, to not drink raw milk, and that's, generally, like, will you be fine if you drink raw milk? Maybe yes, but is there also an equal chance that you will get sick from drinking raw milk? Also likely yes. So don't drink raw milk. Any questions on any of this? Okay. So for the commercial production of milk products, we are starting to not necessarily just rely on these naturally occurring lactic acid bacteria in, these products so these lactic acid bacteria are what are making these acids that help to preserve things. And for commercial production of these milk products, we're now relying on these starter cultures that are added to the milk. And so basically what a starter culture is a preparation of living microorganisms, so these microbes, which are deliberately used to assist the beginning of the fermentation process to produce specific changes in the chemical composition and properties of the substrate. And so the strains of these microbes are carefully selected to produce desirable flavors and textures. And so what's important here is that if you change, the changes in these strains can change the outcome of your product. So different bacteria will cause different flavors and different tastes in your product. And so the starter culture has to be carefully maintained and protected against contamination by other microbes, because then that will completely change the taste and the quality of the food product. So this is cheese production. Cheese can be made from milk of a wide variety of animals, but as you probably know, cow's milk is the most common to make cheese. And so cheeses are classified as very hard, semi-soft, and soft. And so the classification of how hard or soft it is depends on the amount of water content. And so cottage cheese is the easiest to make. And so basically it's pasteurized milk that is inoculated with a starter culture. And then the culture will then cause the milk proteins to coagulate or just become chunky. Also means change to a solid or semi-solid state. And so those coagulated proteins are called curd. And so that's literally, that curd is cottage cheese. It's just that like first step of getting the milk, it's pasteurized, you inoculate it with the starter culture, and you end up getting this like coagulation of the milk, it becomes chunky, and that's cottage cheese. To make other types of cheeses, there's other further processing that's done, and we'll talk about that on the next slide. But so curd is then heated and cut into small pieces to facilitate the drainage of that water or that liquid, and that is called whey. So this is a picture that's illustrating what is happening. And so lactic acid production and renin, we'll talk about renin on the next slide, will cause the milk proteins to start coagulating and becoming thicker. And so then they cut that coagulated mixture to help facilitate the separation of the solids and the liquids so that they can drain the liquids out. So then here they're heated and cut into small pieces. The liquid whey is removed by draining, and the curds are salted and pressed into blocks or wheels for aging. So for most other cheeses, like I said, there's some additional microbial processes that need to happen. And so that is termed really ripening or curing the cheese. So cottage cheese is not ripened. And so there's an enzyme called renin that is protease, it's an enzyme, and it's added to the fermenting milk to help speed up the actual protein coagulation and make it really chunky and thick and harder in content. And so curds are salted after whey is separated and pressed, and then they are ripened to encourage the change in texture and flavor. And so ripening differs depending on the type of cheese you're making. So how long you're ripening it will, be a factor, as well as what organisms you're using to ripen it. So longer ripening will produce more acids, and so more acidic, sharper cheese. And so different microbes can be used to help determine what type of cheese you're left with. So certain organisms will produce certain characteristics. So here there's this Propionibacterium that will result in Swiss cheese. There's this Penicillium roqueforti that you get Roquefort or Gorgonzola cheese. So, different organisms will contribute to this. You don't have to memorize necessarily the names of these organisms, but just kind of understanding, ripening, and that it can vary in time. The longer you ripen something, it's going to become more acidic because you have more lactic acid bacteria for fermenting those products and making those acids, and thus you get different types or more acidic cheeses. So yogurt is just milk that is heated at a really high temperature, and it's inoculated with a starter culture. And so yogurt is usually made from Streptococcus thermophilus and Lactobacillus bulgarius. So there are other Lactobacillus species that can be used to produce yogurt, but the point is that this mixture, the milk heated plus the starter culture, is incubated for several hours at really high temperatures. And so these are just different optimum growth temperatures for the two types of bacteria. So here you see that the S. thermophilus requires a slightly lower temperature than the L. bulgarius. So it's important to know what you're using when you're trying to make yogurt. And so controlled incubation ensures the proper levels of acid production, proteolytic activity, and flavor compound generation. So actually controlling the incubation and knowing the different factors that are needed are important. Any questions on making cheeses and yogurt using milk? Okay. So probiotics, I'm sure you've all heard, are live microbes that confer a health benefit when ingested in sufficient quantities. The key here is insufficient quantities. And so you've probably seen a lot of these over-the-counter types of probiotics. There's so many different kinds nowadays, but it's actually making probiotics is one of the things that's still under a lot of research and study to be able to optimize it for human health. And I'll talk about that a little bit further. But probiotics, the most common ones are Lactobacillus species. This is the most common one that you'll see, as well as Bifidobacteria species. Bifidobacteria is actually associated with breast milk from the mother, and so that's very beneficial microbe that we know of, as well as Bacillus coagulans and Saccharomyces boulardii. So these are the most common ones. There are others that are food microbes that can be used as probiotics, and others that are non-food. Just kind of an FYI here that there are certain strains of E. coli that are not necessarily part of food, but they're still considered probiotics that you can take for various things, which is crazy to me because E. coli can literally range from being commensal to a pathogen to a probiotic sometimes. So yeah, those are some of the common ones that you'll see. Lactobacillus and Bifido are the two that you should know because they are the most common. And if you look at any probiotic bottle, you'll see that there are probably billions of Lactobacillus species or of the same species. So, like I said, the challenge of probiotics is that not all species and strains of microbes are going to have the same activity in the body. So, how do we deliver probiotics to our gut and have them survive getting through the stomach, which is very, very acidic, and it's designed to kill bacteria. If you remember from previous unit, I showed you the gastrointestinal tract, ranging from the stomach, the small intestine, all the way down to the large intestine, and we looked at pH, that wedge of pH, and pH was a lot lower in the stomach, and as you go down throughout the intestinal tract, it starts to become more and more basic. So it increases. And so the reason why the stomach is a lot more acidic is because it's designed to kill bacteria before getting to the large intestine. So this is something that we actually have to think about when we're developing probiotics, because we need the probiotics are live microbes, so they have to actually, make their way through the stomach and not get destroyed by the stomach acids, and then all the way down into the intestinal tract where they are needed. So not all strains and species of probiotic microbes are going to have the same activity in all aspects of their probiosis. That's just the activity of what probiotics do. But one strategy that we do when we know which strains and species to use is having them in large quantities, so they'll have billions of these cells. Another innovation that's used is the type of capsule that's used. It's still an active area of optimization, but essentially there are these innovations where the capsule is designed to have like an outer capsule as well, and then an inner capsule where the live microbes are, so that as it goes through the stomach, maybe the outer capsule will get destroyed, but then the inner capsule can still make its way down to the large intestine and actually get released in the colon where you want the probiotics to end up. So, like with everything though, too much of a good thing is going to be bad for you. So it's not necessarily good for you to overdo it and take all the probiotics possible. It's been shown that taking too much probiotics can cause bloating because of that gas production. So it can cause an upset stomach. Questions on probiotics? Okay, so microbes are also used to pickle vegetables, and for these you don't have to add a starter culture. And so pickling originated as a way to preserve vegetables, and so the cool thing is that pickling uses these naturally occurring lactic acid bacteria that are residing on the vegetables. So that's why you don't need that starter culture, because we're just using whatever is naturally occurring on those vegetables, particularly like cucumbers, cabbage, and others. And so the vast majority of pickled vegetables, including pickles, are now just sold in stores, and they're just simply packed in vinegar, which is acetic acid, to pickle them. So if you didn't know, vinegar is acetic acid. All right, so kimchi is a traditional Korean food, and so this is one of these that uses these naturally occurring lactic acid bacteria that resides on the vegetables. And it's traditionally fermented in a semi-permeable vessel called an ongi, and these ongi are cool because they are these really big clay vessels, but they are special because they breathe. They have these like really, really tiny pores that will allow air to move through the vessel, but not liquid, so they don't leak. And so as the fermentation process is happening, you're getting carbon dioxide being produced, and so it's able to be diffused out of the vessel that allows the reaction to continue occurring because of those pores. So as opposed to putting something in a tight sealed Tupperware, that will limit the amount of fermentation that can occur. The certain amount of fermentation will occur, you'll produce those acids, and that's it. And so using these ongi, you can end up letting it ferment for long periods of time, and once again, and so that allows it to have a very long shelf life. And so once again, the length of the fermentation will change the flavor. So the longer you leave it, the longer it'll become more acidic and change the flavor. So fermentation also can occur through the use of the yeast. So yeasts will ferment sugars and produce ethanol and carbon dioxide. And yeasts are what we use to make a variety of alcoholic beverages, as well as vinegar and bread. Alcoholic beverages include wine, beer, and distilled spirits, if you didn't know that already. And so to make bread, bread rises due to the carbon dioxide that's produced through the fermentation of sugars by baker's yeast, which is Saccharomyces cerevisiae. It's actually a cousin of Candida albicans. Saccharomyces cerevisiae is what we use to make bread. It's called baker's yeast as well, so you should know that. And so any alcohol that's produced will evaporate during baking, and there's also carbon dioxide produced. And so that carbon dioxide is what's going to cause the bread to rise. And so the characteristic flavor of sourdough bread in particular is due to the addition of lactic acid bacteria and potentially other yeast in the place of Saccharomyces to the bread making mixture. So sourdough uses a starter culture. So basically you have the yeast, a living culture of wild yeast, but then you also have that lactic acid bacteria that is used to make the bread rise. So it's created by combining flour and water and allowing it to ferment. And then over time, the natural yeasts and the bacteria that are present in the environment and on the flour will begin to feed on the mixture, and that's how you can make your starter culture for sourdough. So what factors influence the growth of microorganisms in food? So how does food spoil? So understanding food microbiology is essential for maintaining the quality of food and producing it and preserving it. And there are intrinsic and extrinsic factors that will contribute to this. So conditions that are naturally present in the food are intrinsic factors, and then environmental conditions are extrinsic factors. So both of these combined determine what microbes will grow in particular foods and at what rate. Some intrinsic factors are the water availability, pH, nutrients, any biological barriers, antimicrobial chemicals. And so generally, we've talked about this before with just microbes in a lab, in a test tube. They multiply most rapidly when you have moist environments, when you have a lot of nutrients, and the pH is neutral. And so it's similar, for the microbes that are going to be growing on food and spoiling it. So we'll go through a couple of these intrinsic factors. Water availability, foods vary dramatically in terms of water availability. So fresh meats and milk have high water content, which will support the growth of microbes. And so breads, nuts, and dried foods have low water availability. So that's, pretty standard. And so defined populations can grow in these specific environments. Water activity is denoted as AW, and it's used to designate the amount of water available in foods. It's sometimes referred to as osmolarity of a food. And so pure water will have a water activity of 1, and most bacteria will require a water activity of 0.9. And so most fungi require a water activity of 0.8. So like I said, foods, vary in how much water is available. The more water there is available, the more potential there is for the microbes to grow because they like moist environments. And so bacteria need a more moist environment than fungi. Fungi can handle a little bit less water and still grow. Does that make sense? All right, so growth rate versus osmolarity is for different classes of prokaryotic microorganisms. So one of the intrinsic factors that we're talking about is water activity, and so that is used to designate the water available in foods, and the varying availabilities of water can then influence the growth rate of different types of microbes. And so here what I'm showing you is basically a graph. On the x-axis, you have water activity. 1 is the most water, so that's just pure water. This is the growth rate of different organisms and the things that they require to grow. Most bacteria will be, able to grow, obviously, with having the most water content. And then as you decrease the water activity, you have different characteristics of microbes that can grow. So we'll kind of go through what these mean. You don't have to memorize this, but this is a nice table, I guess, that shows you these things. So if I were to ask you, you know, if I were to give you this on an exam and ask you, you know, if I've dried raisins, which is more likely to grow on it, Caulobacter or Staphylococcus? Dried raisins, so very low moisture content. Which one requires less moisture, less water to be able to grow based on this chart? Yes, Staphylococcus, because it's a 0.85. Caulobacter needs a minimum of a water availability of 1, which means you have the most water available. So, does that make sense? So pH is another intrinsic factor. Obviously, we talked about this. It's important in determining which organisms can survive and thrive on specific foods. So many microorganisms are inhibited by acid conditions. Like I said, the stomach will try to kill microbes because it's very acidic. But lactic acid bacteria are special in that they are not really inhibited at low pH. And that's why they're used in fermentation for food production and preservation. But lactic acid bacteria, because they are producing these acids, they can also cause spoilage of unpasteurized milk and other foods. And so yeast and other fungi can survive at relatively low pH, so more acidic pH. And so most acidic foods will spoil from fungal contamination as opposed to bacteria, because the acidity will kill the majority of microorganisms, and so fungi can counter that. They're sturdier, they're hardier, and so they can survive more acidic environments. So if I were to give you an example of a food that spoiled, and I were to ask you, was it more likely to do with fungal contamination or bacterial contamination, you would tell me it's fungal, because you now know that fungi can handle and survive at relatively low pHs or acidic environments. Any questions on any of that so far? Yeah, absolutely. So acidity or acidic environments, that just means you have a lower pH. And so if you have a more acidic environment, we know that bacteria usually are killed by certain acids, with the exception of lactic acid bacteria, because those are used for fermentation. But many of the microorganisms are inhibited by acid production and conditions. And so an example of that is bacteria in the stomach. Usually there isn't a lot of bacteria in the stomach because they're killed off because the stomach is very acidic. And so on the other hand, though, yeasts or fungi, they are eukaryotic. They're not prokaryotes like bacteria. They have chitin in their cell walls, so they're stronger and they're sturdier, and they can actually survive at relatively low pHs, so more acidic environments. So this is a pH scale from 0 to 14, going from most acidic to basic. So most acidic foods foods that are acidic in nature, because we know that microorganisms can't grow as much on acidic conditions. If you see acidic foods spoiling, then it's probably from fungi and not from bacteria, because the fungi can survive the low pHs of acidic environments. Does that make sense? Yeah. I'm happy to reiterate any concepts for you guys. So some other intrinsic factors are nutrients. So obviously, nutrients present in the food will determine which organisms can grow. So if you have a lot of sugar, that's going to allow a lot of different organisms to want to feed on that sugar. Biological barriers, so rinds, shells, and other outer coverings help to protect the food from microbial invasion. But eventually, the microorganisms that are naturally occurring on those fruits, for instance, will break down those coverings and cause spoilage. A good example of this is like leaving your apples really long out, or even like your pumpkin, leaving it out on your porch for a really, really long time. They can last for a really long time because they have this outer protective shell. But then over time, if you leave it for, several months, you'll notice that, it's definitely rotting and mushy and gross and spoiled, and that's because of those microorganisms. They eventually are able to break down that outer covering of the pumpkin and cause that spoilage. There are also antimicrobial chemicals that are produced. So some foods and spices, like lots of herbs and spices, naturally have these antimicrobial chemicals that inhibit the growth of organisms that are responsible for spoilage. So that's why, if you have a lot of herbs and spices in your cabinets, you can use those for a very long time. They don't really rot or spoil. Think about it, they're very dry, so they don't have a lot of water content because microbes thrive on water. But then also they have these antimicrobial chemicals that will inhibit the growth of organisms that allow them to spoil. When I talk about intrinsic factors, you can have things that, if you have more nutrients, then that can cause more organisms to grow, but then these intrinsic factors can also be protective in the case of having the biological barriers and having these antimicrobial chemicals. So extrinsic factors are, the factors that are obviously that are not intrinsic, so they're not within the food item. So basically, the extent of microbial growth will depend largely on these outside factors that are like how you store your food. And so microbes multiply rapidly in warm, oxygen-rich environments because they like, moisture and they like humidity, and so they really like that. So microbes will also grow under low oxygen and anaerobic conditions. So if you have canned foods, that's trying to, if you have a canned food and it's not properly, sealed, you're trying to maintain the amount of oxygen that can go in there because it's sealed, it's pressurized, and it's anaerobic, because there isn't much oxygen coming in. But if it's improperly canned, then that can definitely still allow microbes to grow. So basically my point here is that microbes will multiply more rapidly in warmer, oxygen-rich environments, but there's always an exception to the rule in the case of like having canned foods that are not properly canned. And so some extrinsic factors that are most important are the storage temperature, oxygen levels, and humidity. Can anybody think of where these factors, like the storage temperature, levels of oxygen and humidity, are like so important to maintain, to keep those things really, really like maintained? Like what type of environment, where would we want to make sure that we don't screw any of these up, because then you could screw up the whole like system. Kind of an open-ended question. Like restaurants and bars, and this is kind of like a weird question to ask, but like, yeah, so like if you have restaurants and bars, they're gonna have like a walk-in freezer that, the temperature is really maintained, but and like walk-in fridges as well. But on top of that, they really have to maintain the oxygen levels and the humidity, because if they don't, you're gonna literally contaminate and screw everything up. And so restaurants are really, difficult to maintain because of that. So that's where these extrinsic factors are most important. So storage temperature will affect the rate of microbial growth. Obviously, if you have lower temperatures, so the colder it is, you're going to have very slow growth of microbes or even non-existent microbes, especially if you're, freezing things. And so freezing things will result of inability of microbes to grow. So like right now in the lab, when I have my cultures and I, will make, something grow up, and then I want it to kind of like stop growing, I put them in the 37 degrees C incubator. And so the fridge is 4 degrees Celsius. And so when I want your plates to grow, I'll put them at 37 degrees for 24 hours, and then I'll move them into the fridge because it's colder, and so it can kind of slow the growth of them. They still grow because it's the fridge, but if I want to completely halt their growth and actually like just preserve them in the state that they are, kind of creating like zombie cells, you freeze them. So that's why, freezing food allows it to last longer. So some of these concepts are inherent. But the atmosphere as well is really important, not just temperature. So the presence or absence of oxygen, like I said, will affect the type of microbes that can grow, because some of them, obviously more microbes will grow in the presence of oxygen, but there are anaerobic organisms that can grow without oxygen, or some that are able to grow in low oxygen levels. Humidity also, humidity is correlated to moisture content, and that will promote the growth of spoilage microorganisms as well. So if I give you a scenario that says I have a high oxygen, high humidity, restaurant, fridge, and but it's in a fridge, so we have low temperatures, but there's high oxygen, high humidity. Is it more likely to have microorganisms growing or less likely? More likely, because you have more moisture and more oxygen, and organisms thrive on that. Any questions at this point? Okay, so how can food be preserved longer? We talked about how it can be spoiled. Now, how can we preserve it? The goal is to prevent the growth and limit metabolic activity of microbes that cause spoilage and foodborne illnesses. So now we're talking a little bit further than just spoilage, but now also foodborne illnesses. So preservation methods will help, prevent illness. And so preservation methods interfere with those intrinsic and extrinsic factors. And so some of these include canning, pasteurization. We've done pasteurization before in lab with the pepper experiment in the very beginning, where we put it in a water bath for 5 minutes, and just to kill off, the majority of what's growing in there, not necessarily sterilizing it. So we'll talk about differences between pasteurization and sterilization. Cooking obviously preserves your food. Refrigeration, freezing, and then drying things or reducing the water availability will also limit the activity of microbes that are able to cause the food spoilage and illness. So when you cook, you can destroy organisms that don't form spores. So some organisms can survive, if they form spores, then the spores are like a hardy component of the organism that allows it to kind of still germinate even under really high temperatures. So cooking can destroy non-spore-forming organisms. Obviously, it alters the characteristic of the food, including taste and just texture and even smell and all of that. And so if the heat is uneven on your food, then some organisms can still survive in the undercooked portion of the food. So that's why it's important when you're, making a steak, if it's like frozen and you took it out to thaw, the very inner part of it might still be more frozen than the outer portion. So it's important to make sure you heat your skillet like really well, fully, and then try to not have it under really high heat, under like medium low, so that you end up, having the heat be able to spread throughout the entire portion of the food, so that the more undercooked portions aren't, so that there aren't organisms in those undercooked portions. So refrigeration obviously will preserve food because it slows the growth rate of spoilage organisms. So if I were to ask you a true or false question, refrigeration preserves food by inhibiting the growth rate of spoilage organisms. Would that be true or false?
You say it again.
Okay. Refrigeration preserves food by inhibiting the growth rate of spoilage organisms. False, because it doesn't inhibit it, it slows it. So those are things to make sure you keep straight in your head. And so yeah, because some organisms still are able to multiply in low temperatures, so that's really important. Freezing, though, will stop microbial growth because your water availability is unavailable because you're forming ice. And so the portion of organisms that remain in the food can grow only when it's thawed. So that's why for certain foods you're not supposed to freeze thaw, because that can definitely cause the growth of organisms. Sometimes there's certain foods that are packaged and they'll say only thaw before use and don't refreeze it. So drying and reducing water availability obviously will inhibit microbial growth because it's decreasing the available moisture. But you can still have molds that grow, so molds will be more fungi related. And then irradiation, there's some fruit that are irradiated, and basically irradiation is basically damaging microbial DNA through like UV light. Okay. Any question on food preservation methods? So what about liquids? How do we, preserve liquids? There are two ways that we'll talk about. There's filtering liquids, like we've done already, where we filtered water, and so we were, trapping the bacteria on the filter. And then there is heating. You can heat liquids, obviously. So filtering, we've talked about this in lab. The filter, the microbial cell filtering usually uses filters that are 0.2 micrometers or microns in size. And so that, will not remove smaller things than 0.2 microns like viruses and other small molecules. And so heating will obviously, kill a lot of microbes, but it really, the efficacy of heating depends on the temperature and how long you heat for. And so sometimes you can boil things, but certain organisms that form spores aren't killed because they form those endospores, and so boiling sometimes doesn't even kill those. So there's got to be ways to account for killing everything, even the ones that are more hardy and sturdy and can survive heating and boiling. Pasteurization, we've done in lab before. It's the process of heating a liquid for a specific period of time to destroy pathogenic microorganisms. So it doesn't necessarily destroy everything that's in there, but you're heating a liquid for a specific time. This was developed by Louis Pasteur. The name comes from his last name, in 1864 to improve the storage quality of wine. And so commercial pasteurization, so pasteurizing milk in just, commercial aspects for production began late in the 1800s in Europe and in the early 1900s in the US. So the process of heating or boiling milk for health benefits has been recognized since the early 1800s. This was used to reduce milk-borne illnesses and mortality in infants, especially in the late 1800s. 1 in 4 infants was, really dying in the 1800s based on milk-borne illnesses because they weren't pasteurizing the milk and killing the organisms. So as society industrialized around the turn of the 20th century, increased milk production and distribution led to outbreaks of way more milk-borne diseases. And the common ones at that time were typhoid fever, indolent fever or brucellosis, scarlet fever, septic sore throat, that's strep throat, diphtheria, and diarrheal diseases. Tuberculosis also can be transmitted via milk that's not pasteurized. So now milk is obviously required to be pasteurized before it can be produced and put into our stores. In 1918, this lady, Alice Catherine Evans, was able to show that drinking unpasteurized milk could transmit the bacteria that cause brucellosis, so Brucella abortus, from domestic farm animals to humans. So that's why pasteurization became mandatory for all milk sold within the city of Chicago in 1908. And in 1947, Michigan actually became the first state to require that all milk for sale within the state had to be pasteurized. So Michigan's always been one of the leaders in things like that when it comes to health. So in combination with improved management practices on dairy farms, milk-borne illnesses were virtually eliminated with the commercial implementation of pasteurization. So we started to pasteurize milk and it pretty much virtually eliminated, these milk-borne illnesses. In 1938, milk products were the source of 25% of all food and waterborne illnesses. And then in 2016, they accounted for far less than 1% of food and waterborne illnesses. So we're doing something right. So some types of pasteurization, that we'll talk about, because I kind of vaguely said, okay, you're having a high intense treatment for a certain amount of time. So what does that really mean? There are different types of pasteurization. So one example is ultra pasteurization, where you're basically putting the milk at 140 degrees Celsius, that's 284 degrees Fahrenheit, for context. And so you're just doing that for 2 seconds. You're holding the milk at that really, really, really high temperature for 2 seconds, and then you can just put it in the refrigerator and it can be stored and you can have extended storage. There's another technique called ultra heat treating, or UHT, and so that process will hold the milk at that same temperature, but for 4 to 15 seconds. So it's the same temperature, but a longer period of time. And so when you do this ultra heat treatment, this actually sterilizes the milk, and it's not necessarily pasteurized. So sterilizing is even going further and completely, like, you're sterile, there's absolutely nothing in there. And so this process lets consumers to store milk for several months without even refrigerating it, just because of those extra few seconds in the 140 degrees Celsius. Questions? Okay. So these are some examples of types of pasteurization methods. So 63 degrees C for about 30 minutes, that's regular pasteurization. When we did it in the lab, we did it at about 63 degrees for, I think, 10 minutes. So that was just because we were using a small pepper sample. And then, you can have either high temperature short time pasteurization, or HTST. So that's high temperature, 72 degrees Celsius, short time, 15 seconds. You can have higher heat, shorter time pasteurization. That's even higher heat in 1 second, shorter time. Another one of those would be even higher than that. So 89 degrees Celsius, then 90 degrees Celsius for either a second or half a second. You can even do this further for literally 0.1 seconds or 0.05 seconds just by raising it by a few degrees. And so these are just examples of that. And then ultra pasteurization is what I just talked about on the previous slide. So this is, 138 degrees Celsius for 2 seconds. And then the second one I talked about, which was the one that sterilizes it, was the ultra high temperature process. So 140 degrees, but for 4 to 15 seconds. Yeah, so there's like autoclaves in certain machines that will do that. Yeah, it's really, really high. So what else is pasteurized? So not just milk, there's juice that's sold on the shelf at room temperature. It has to be sterilized because it's able to sit at room temperature. So remember when I said that the ultra heat treating will sterilize things, and so this lets them store milk or juice for several months without refrigeration. So that's how you store juice at the counter, at the shelf, at room temperature, because it's been sterilized. So no matter what it says on the container, if it says it's pasteurized, it's not true, it's been sterilized. If it's been pasteurized, don't drink it. Usually this is done by ultra heat treatment. So all truly pasteurized juices still need to be refrigerated because they're pasteurized, obviously. And so many different products sold in the supermarket may be pasteurized besides milk, including fruit juices, almond cider, and some beers. And so can anybody guess why, if you were to take, juices stored at the shelf, but once you open it, you have to refrigerate it? Can anybody tell me why, when you open it, what is there access to? Yeah.
You're exposing it to oxygen.
Yeah, you're exposing it to oxygen. And then also the environment, so microbes can come in, and so that will then allow the growth of microbes. So that's why you have to refrigerate things once you open them. So we'll end here. So what does sterilization mean? It refers to any process that completely eliminates, removes, kills, or deactivates all forms of life and other biological agents like fungi, bacteria, viruses, any bacteria that form spores, prions, unicellular, eukaryotic organisms, like everything. So that's present in that specified region, like a surface, liquid, medication, or a compound such as biological culture media. So this is literally the Wikipedia definition that just encompasses it all. So it's important to know the differences between what pasteurizing does and what sterilization does. And so next week we'll continue on with the rest of this lecture and talking about does boiling a liquid actually sterilize it, and we'll go from there. Any questions so far?
Why don't they just all treat like all milks then? Like we'll have to put them on the shelf first, because it's the same temperature as when you're pasteurizing it. So.
Yeah, I think that's a good point, but I think it also has to do with like preserving the quality of it. So you don't want to sometimes the microbes are utilized to continue to ferment in there and give you like a taste. So the microbes are also still good. You don't want to completely like kill everything off. So that is probably why you want to still have those health benefits. But yeah, that's a really good question. Yeah, people still have to take it, so I'll be putting that up.
2 weeks or 5 more.